Hello everyone, my name is Shweta Goyal and I'm presenting my PhD thesis work as a part of Saransh competition. So first we have to know that what is bisphenol A, that is BPA. BPA is generally the main component of the plastic consumer products and used as a food container, water bottles, and also used as a epoxy resins in the inner lining of the metallic jars and also represented as a 7 resin coat. So what are the BPA major concerns? On the high pH and temperature, BPA leaches out inside the hot food and water that consumed by the person. On the high demand of the BPA products nowadays, it is expected that its growth rate will be increased by the 4.7% in the next coming years. So there are several studies which has reported that there are various diseases due to BPA exposure, including the neurobehavioral changes, which causes neurotoxicity and leads to learning and memory dysfunction. So what are the rationale of our study? In the living organism, there are different kind of living cells and including the neurons. Neurons is the basic fundamental functional unit of the brain. Hippocampus is the major region of the midbrain which is responsible for the learning and memory. In the living cell, nucleus contains DNA and mitochondria which fulfill the energy demand and they communicate for the successful mitochondrial formation, that is mitochondrial biogenesis. In this process, PGC1 alpha encodes more than 99% proteins of the mitochondria and which imports inside the mitochondria via the import machinery, via G4 mediated import machinery. And after the successful mitochondrial biogenesis, mitochondrial fission and fusion maintains the mitochondrial number where the DRP1 is an essential factor for the fission. If there is an impairment, neurodegeneration causes the different neurodegenerative disorder, including the Alzheimer disease. And what is the aim of our study? That how these processes are impacted by the BP exposure in the rat hippocampus, which causes neurodegeneration, and how can the therapeutic approach can be effected against this BP exposure. So in our PhD work, we found that BP impairs the PGC1 alpha level in comparison to control, while the DRP1 level elevates in the BP exposure and impairs the G4 level, which imbalances the mitochondrial biogenesis and dynamics, which triggers the cytochrome C release from the mitochondria and elevates caspase level and impairs the learning and memory in the rat. So as the therapeutic approach, when we overexpress the G4, we found that G4 expression can repair the mitochondrial biogenesis, while the PGC1 alpha overexpression by the nicotinamide can balance DRP1 and G4 level, which can inhibit the apoptosis and restore the learning and memory function of the rats. Please avoid the 7 number resin code BP products. Thank you.